Hello and welcome to Ara and Friends. And today we're going to be with our Ara team and we're going to go inside the lab. So at the beginning here, we're going to introduce everybody. We're going to talk about microbiology and we're kind of going to go back to the basics. So I'm going to let our team introduce themselves. Megan, let's start with you.
Hi, I'm Megan Hicks. I am the lab director here at Ara Testing. I have been in this dietary supplement industry for 13 years, and I've worked with a lot of people that are probably going to be viewing this, and I'm excited to show you the lab and how we do our methods here.
All right, Tommy.
All right, I'm Tommy Geisendorfer. I've been in the lab setting about 6 years. I started out as just a typical chemist, microbiologist, analyst. Worked my way up into quality, and now I'm the quality manager here at Ara Testing Labs. And we're going to show you some micro testing procedures today.
The first segment we're going to watch is Megan's going to go in the lab and show us the initial steps and the weigh-ups and different sample matrix that she does. Let's watch and see what Megan shows us.
I'm going to be weighing up a sample that needs full USP methods and micro. That's got all of them.
Okay. So you're going to weigh it once and try and pull for APC, yeast and mold, coliforms, E. coli, salmonella,
and BTG, yes.
And Entero. Okay.
So these are little sterile weigh books.
Okay, so what kind of product is this?
This is capsules, so it's a normal matrix for us. We see a lot of capsules, powders, and tablets in our lab. Next step will be to pour our TSB broth into the sample and stomach it until it's homogenized. Capsules run for 60 seconds usually. Okay, so now we're taking our stomacher out of, or sample out of the stomacher, making sure it's all homogenized, all the capsules are broke open and emptied out, which it looks good. Then we'll make our dilutions and put them to the appropriate petri dishes. They are labeled with the sample number and dilution on them. My second dilution, just inverting, make sure it's mixed to make my third dilution. And then our sample bag, the stomacher bag, will be clipped and put into the incubator overnight to proceed to the test tomorrow as E. coli, salm, staph. We're going to pour our agar for the USP method for aerobic plate count, which is, we use TSA, tryptic soy agar. So we'll pour our sterility and our samples and swirl them and let them solidify before putting them into the incubator.
What's the next agar?
This is for yeast and mold. This is an SABC. It's a acetylodextrose agar with a chloramphenicol. Now we're going to do our coliform USP auger, which is VRB violet red vial auger.
Okay, we're back in the lab with Megan. Tell us what you're doing now, Megan.
I have an environmental swab for my customers, a sponge swab down in here. And I'm going to pour my broth in, get it mixed in really well, and plate Entero Petri film again.
And what's your broth?
TSB. Mix it up.
And this is a sponge, like, you know, probably an equipment sponge or something for that plant's environmental testing.
Right.
And on this one, do you do dilutions 1 and 2?
Yes, 1 and 2. So 1's out of the bag and then 2's out of the peptone tube. I made a dilution.
Thanks for showing us Entero.
This is an environmental sponge swab from a production facility, and we're gonna do a salmonella AOAC method on it. So we're gonna put our tryptic soy broth in here and let it incubate overnight. And then tomorrow we will do a 1-2 test on it, which is AOAC approved. My broth in, and then I'll mix it up and get it in the bin to go into the incubator till tomorrow.
There's a lot of different methods that you can use in you know, our standard negative microlab. And today we've talked a little bit about USP, but also in the video you're going to see Tommy do some AOAC and Megan. They both did some AOAC methods. Tommy did an AOAC salmonella method, and Megan did an AOAC Entero Petri film method. So one thing we want to do is just kind of overview the AOAC methods as well, because there's another choice, and sometimes they're faster, but faster isn't always the best whenever it comes to a complex matrix. And I'll let Megan kind of take it from here on why we would, what we would need to do to choose AOAC.
Yeah, so Petri film is a little bit trickier with different pHs and matrix. It can cause interferences on those plates, so we aren't able to read them as accurately. So to do a Petri film on a new product, we like to really do a validation. We like to spike it, make sure we can grow what we're trying to test for in the lab. So we did an Entero on a environmental swab, which is a very neutral matrix. So that one typically works great, but Petri film can be a real picky plate for different matrices, so we always try to do a validation to make sure we're growing out everything in that sample, where the USP methods are a little bit more forgiving.
And I think the USP, though, we also, encourage everybody to go ahead and spike, use their sample, spike the positive to make sure that their matrix can grow. We have seen a few issues with probiotic samples on even USP methods. So just always doing that spike, that control sample to make sure that you can grow that organism, no matter what the matrix is, very important. But like Megan said, on AOAC, there's so many interferences that, it's pretty much, we wouldn't want to do that method without some verification, validation that your product could grow on that. Okay, Tommy, on AOAC, what's your take on some of the tests that we do?
They're great for doing rapid testing. They may not be as comprehensive or matrix forgiving as USP, but they are a lot more rapid, especially for salmonella. Salmonella, we have a 1-2 test. It's basically an L-shaped tube with 2 caps on each end. You put in a reagent on each side and then your sample, and it tests if there's motile salmonella that moves up into this it's a antibody, and it makes like an immunoband, and it cuts down a day off your USP salmonella testing, and it's pretty forgiving as far as matrices go. And it also works with 10-gram or a 375-gram sample, so you can do as big a sample as you want if you're doing large scale food testing or anything like that. Um, and as far as Petrifilm goes, like I say, it cuts down on your testing time by usually a factor of a day or two, which is great for people that need rapid turnaround. And they can be validated on a wide variety of foods or supplements or whatever matrix you have.
Hi, Tommy. What are you working on today?
We're going to do some salmonella 1-2 tests. It's just a presence-absence test for salmonella. We'll take a incubated sample, incubated overnight, TSB. We're going to put in these tests and see if it has salmonella.
All right. Okay, so this is the kit.
Well, this is the kit. So, step 1, I'm going to take off all these black caps. Alright, first step, keep your iodine, put it in the black cap side. Just 1 drop a piece. And with this little funnel, ensures your iodine gets down into your 1-2 test all the way. Black cap back on. Next step, remove the white cap side. We're going to get 1 drop of the antibody reagent on the white cap side. Take our fancy clippers and clip off this tip portion of the cap. Okay. Now these tests are ready to go. Then you will inoculate them with 100 microliters of your sample into the black cap side, and then incubate them 16 to 24 hours in a 37-degree incubator. Alright, so these swabs have been incubated overnight, and now we're going to take 100 microliters of them and transfer them into the 1-2 test portion. So this is going to be day 3 of the E. coli USP test. So we weighed the sample, incubated overnight, day 1, and TSB, and then we transferred day 2 to Mac Roth, incubated in a 40C incubator overnight, and then day 3 is today, and we're going to streak to Mac Auger, and then again incubate overnight for consumption. This sample was incubated overnight in TSB, and now we're going to transfer it to MacRoth to do the USP E. coli test, which to RVS broth, which is USP vanilla. We're going to streak it on mannitol, and that's USP staff. So that's what we're going to do right now. It's going to be a mil and an inch to it.
The thing that we really didn't video and that it's a little bit boring is we're hoping in a clean micro setting, and then when manufacturers are manufacturing, their samples are clean. So a lot of times we're just counting blank plates. And so, they're called negative micros for a reason. There's nothing on there. So we didn't videotape, very much of the actual reading because it's just reading, zeros a lot of times. So we'll probably do another video, maybe even next month, and just keep this video going and do a little bit of a probiotic testing overview. While I've got this team on the call, I thought we would talk about the USP update that went into effect April 1st. So this was a great month to have a standard micro overview, and we can kind of talk about the updates to the USP. We do a lot of dietary supplement testing, so we're testing with USP 2021 and 2022, every day, all the time. So, Megan, you want to take it from here and kind of just give like highlights of the changes that the USP put in?
Yeah, so there's two major changes for us that we noticed is the aerobic plate count method. It has increased the incubation time. It's still 30 to 35 C, but it's now went from 48 hours to 3 to 5 days. So 72-hour minimum incubation now. And then on the E. coli test, the plates now can be pulled at 18 hours to 24 hours. And so that's a little bit shorter incubation time for us.
All right. I knew that the updates seemed to be, wasn't a huge amount of changes, but then the APC does impact, at least one more day of incubation. So when people are used to getting their APC results in 2 days, it'll be a minimum now of 3 days. So that does kind of impact the manufacturing a little bit. Well, thank you for joining us, and thanks for Megan and Tommy being on. And I just appreciate, I know whenever we go in the lab, it makes them slow down and I interrupt their day. So I appreciate them letting us go in the lab, see what they do, and for them showing us, some behind the scenes on some testing. So thanks. We'll see you guys again. Bye.